Good morning, everyone. Welcome to our webinar. I'm Gaurav Agrawal, Director of Scientific Applications at Eurofins DiscoverX. Thank you to Xion Laboratories for hosting us. The title of today's webinar is Cell-Based Assay Solutions for Peptide and Small Molecule Therapeutics Targeting Obesity. Accelerating Screening to Late Phase Development. So allow me to begin by highlighting the profound impact of obesity on global health. The data unmistakably reveals that the obesity has escalated into a worldwide epidemic, with projections indicating that global obesity rates could soar to 25% by 2035. So the urgency for next generation obesity treatments has never been greater. Therefore, it is critical to develop effective and accessible therapeutics for obesity as soon as we could. So despite its significant impact on human health, it's only been in the past 4 years that the therapeutic development landscape for obesity has gained substantial momentum, as evident from the therapeutic development timeline. Now, this progress can largely be attributed to the FDA approval of the GLP-1 therapeutics, such as semaglutide, marketed under the brand name Wegovy by Novo Nordisk. It is quite apparent from the clinical program landscape that you see here that the GLP-1 is a predominant target for most of the obesity therapeutics in development. Now, following semaglutide, we have seen the introduction of next generation dual agonistic peptide therapeutics, Tirzepatide from Lilly, marketed as Zepbound. Now, it is a unimolecular dual peptide agonist for both GLP-1 and GIP receptors. Fueled by these groundbreaking approvals, there has been a remarkable surge in clinical programs over the last 4 years, and numerous dual and triple agonists, peptides targeting GLP-1 and GIP and glucagon receptors have been developed or under clinical development, making a significant advancement in the obesity treatment research. With the remarkable growth in the clinical programs focused on obesity, the market size has already surpassed $24 billion in 2023, boasting an impressive CAGR of 27%. Projections suggest that the market could exceed $130 billion by 2028. Currently, there are over 80 unique assets in clinical development, both for peptide and small molecule therapeutics, with more than 600 programs in the global pipeline. Leading biopharmaceutical companies, as you see here, include Lilly, Novo Nordisk, Amgen, AstraZeneca, Pfizer, and others. And they are all actively contributing to this dynamic landscape. From the graph here, you can see the market projections for obesity market are certainly influenced by various factors, including additional approvals for treating conditions related to obesity. A key one of that is cardiovascular diseases. Now, the duration of treatment regimes and global patient access to medications will also influence the market size, but nonetheless, it remains a highly promising avenue deserving our global attention. Now, before I move forward, I'd like to provide a quick introduction to Eurofins DiscoverX and how are we helping this therapeutic landscape in accelerating obesity drug discovery. So DiscoverX is a leading company in the industry with the biggest assay portfolio of cell-based assays, with over 900 assays for over 10 plus druggable target classes. But we are particularly known across the industry for our keen focus on GPCR drug discovery. We have over 25 years of experience developing MOA-reflective functional cell-based assays for GPCRs for both small and large molecules. On this slide, I'm particularly focused on our capabilities for obesity therapeutic development. We have over 50 publications, patent filings, and mentions in the regulatory filings from industry-leading companies like Lilly, Pfizer, and Amgen. We are already supporting 25 plus global potency testing programs in multiple geographies with a comprehensive assay portfolio for all the key targets such as GLP-1, GIP, glucagon, and now amylins. We have went an extra mile and have fully qualified our assays for FDA-approved drugs such as Ozempic, and more recently with Tirzepatide, and have collected a comprehensive data set to show our assays meet stringent regulatory requirements when implemented in GMP lot release testing. We do offer all relevant MOAs. You will need to establish accurate pharmacology for your therapeutic, including cyclic AMP, beta-arrestin recruitment, and GPCR internalization assays. And to make sure our assays are phase appropriate, we provide them as stable cell lines, thaw-induced Bioassay kits, and working cell banks. So this is in a nutshell capabilities at DiscoverX. Now let's look at what are the key targets that are currently being investigated for developing obesity therapeutics and how cell-based assays are critical for their development. So the three key targets that are of prime attention, as you see here, are GLP-1, GIP, and glucagon. All are GPCRs, where several dual and triple agonistic peptide therapeutics and small molecules targeting these receptors are in various clinical development phases. Now, for developers to take their programs through the drug discovery pipeline from screening, characterization, and all the way up to late phase clinical development, they need MOA-based assays. Since all the key targets are GPCRs, the key MOA, unsurprisingly, are cyclic AMP accumulation assays, beta-arrestin recruitment assays, and since receptor recycling and desensitization are quite important to study drug efficacy, internalization assays for these GPCR targets are quite important as well. Since GPCRs could elicit multiple pathways when activated, such as G protein dependent, second messenger like cyclic AMP, or recruits beta-arrestin followed by internalization, there could be multiple effects of the drugs, some beneficial while others not so much. Hence, developers often leverage introducing ligand bias in their therapeutic design to select candidates that align or basically retain signaling through the beneficial pathways, which shows a bias against other undesirable signaling events. Thus, the availability of cell-based assays for evaluating various such pathways is highly desirable to select the most appropriate candidate. Here I will showcase an example in the next slide. Now, of course, obtaining and reporting such characterization data is critical for regulatory filings such as IND and later in your clinical phases. Therefore, the cell-based assays are also critical for clinical development phases such as evaluating neutralizing antibodies and subsequently for quality control during manufacturing to support the commercial release of therapeutics. All right, so let's take a look at how cell-based assays are leveraged for the development and characterization of biased therapeutics with desirable pharmacology. So here is a quick case study where I'm showing an unbiased agonist for GLP-1 receptor as shown here. This particular ligand, when binds to GPCR, stimulates both cyclic AMP and beta-arrestin pathways equally because it is an unbiased agonist. Since recruitment of beta-arrestin triggers accelerated internalization, we see a reduced insulin secretion profile and shortened action duration and tolerance development to the therapeutic that eventually leads to the drug being less effective. So however, now you see on the other side, let's review a properly developed biased therapeutic such as Tirzepatide, which is a dual GLP-1 GIP dual agonist. Now this molecule is developed to be biased towards cyclic AMP pathway, as you can see here, and have a minimum activation of beta-arrestin recruitment to GLP-1, while it remains a fully unbiased agonist to the GIP receptor, as you see on the other figure. Now what you see here, the Tirzepatide clearly shows advantage over unbiased agonist in having an enhanced insulin secretion profile, reduced desensitization, and shows prolonged receptor activity. We have collected equivalent data to establish this profile, as you see here, with our cell-based assays. Figure A and B shows cyclic AMP and beta-arrestin recruitment assays for GLP-1 receptor. It is evident that Tirzepatide in the teal color here is equally efficacious as the unbiased GLP-1 agonist Exendin 4 in the cyclic AMP assay. However, it shows a significant bias against recruiting beta-arrestin in the figure B. It is also notable that the cyclic AMP assay, that the potency of Tirzepatide is multifold lower versus Exendin 4, and this is deliberately done to tune down the GLP-1 agonism to minimize side effects, while Tirzepatide remains a full and unbiased agonist to GIP, as evident from the figures C and D. Hence, making this drug an imbalanced biased dual agonist, Tirzepatide has so far shown a lot of clinical promise as an obesity drug, with over 13% weight loss in the population. From this case study, it is well evident that the cell-based assays, particularly cyclic AMP and beta-arrestin, are quite valuable tools for the proper development and characterization of such complex therapeutics. And in fact, the beta-arrestin assay that I've been showing you here, it is used from DiscoverX and by Lilly in their characterization of Tirzepatide, and is mentioned in the regulatory filings and publications. In addition, as you can see here, several other developers globally are using DiscoverX assays for obesity therapeutic development, and over 50 plus publications we have in patent filings that are showing characterization data sets with our cyclic AMP and beta-arrestin assays and internalization assays, both for peptide and small molecule therapeutic development. Now let's quickly look at our technology platform that is utilized to build all our GPCR and 800 plus other assays other than GPCRs. We call our technology enzyme fragment complementation technology. Now it is based on a split beta-galactosidase system where the enzyme is split into two inactive fragments called ED or enzyme donor and the EA or enzyme acceptor, as you can see on the left hand panel. Now these fragments can be attached to different molecules in the cell that come together under the right stimuli, such as addition of a ligand, peptide, or a small molecule to GPCR, for example. Or you could also add these fragments stepwise exogenously attached to certain molecules to cell extracts or to set up competition assays, as in case of cyclic AMP that you see, where the ED tagged cyclic AMP competes with cellular cyclic AMP produced as a result of GPCR activity. Now more cellular cyclic AMP is produced, more it outcompetes the ED tagged cyclic AMP in binding to the antibody, which is also added to this assay. This complementation puts together a functional beta-gal enzyme, and this complementation activity is highly quantitative as a chemiluminescence signal produced when the enzyme hydrolyzes a substrate. The cyclic AMP assay is a gain of function assay and typically produces a large assay window, as you can see an example curve here, and produces a sensitive detection. Now cyclic AMP assays could be used for several purposes, but primarily for potency testing, ligand bias, accessing albumin potency shift, NAP testing, and GMP lot release. So cyclic AMP assay remains as a main MOA for potency testing for multiple programs that we are supporting globally. Because cyclic AMP signaling is an unbiased signaling compared to the beta-arrestin, and hence it remains the key assay for potency testing. Now, our assays are homogeneous, sensitive, robust, and they have a luminescence readout, as I was telling you, and they're easy to transfer. So all these characteristics makes our assays easy to work with, easy to perform, producing a robust assay window, and you can transfer these assays across multiple sites because they are easy to run in the lab. Moving forward, quickly covering the other two key MOAs, beta-arrestin and receptor internalization. In beta-arrestin assays, the GPCR, for example, GLP-1 is tagged with our ED tag, and beta-arrestin is tagged with the larger enzyme fragment, the EA tag. In presence of the stimuli, or in this case GLP-1 or any of the peptide analogs, the GPCR recruits beta-arrestin, bringing together the EFC fragments, and hence the complementation happens. You add the beta-cal substrate to the cells after lysis, and it produces a robust light signal. Similarly, in receptor internalization assays, we also leveraged the enzyme fragment complementation technology, where the GPCR is tagged by the small ED tag. Now what we have done, we have tagged the endosome with the other EA tag. When the ligand binds to GPCR, for example GLP-1 again, the GPCR is activated and is internalized. Now this internalization puts the GPCR in the endosomal compartment, where it combines with the other EA fragment of the beta-gal, and this provides a complementation of the enzyme, and again in presence of the substrate produces a robust light signal. Now both beta-arrestin recruitment and receptor internalization assays can be used to establish precise mechanism of action for your peptide therapeutic or a small molecule therapeutic, evaluating signaling specificity, establish ligand bias as I showed in the last slide, compare potency shift with native ligands, and confirm biosimilarity. So these are the key utilizations of beta-arrestin and receptor internalization assays for obesity therapeutics. Moving forward, I would like to touch upon the target receptors, what we have, the key mechanism of actions we have developed, and the assay formats we have developed for each of these targets that makes them easy to access to our clients. So for all GIP, GLP-1 and glucagon, we have developed cyclic AMP accumulation assay, beta-arrestin recruitment assay, and receptor internalization assay as the mechanism of action assays. And they're available as stable cell lines, working cell banks, and bioassay kits. So these are off-the-shelf products and easy to access through ordering from our website or through our business development team. Now let me spend a little bit time to talk about different formats that I just described in the previous slide. Mainly the stable cell lines, the working cell banks, and Thorne-used qualified bioassays. Now what you see here, in the first panel we have the stable cell lines. These stable cell lines are shown to capture accurate pharmacology with high passage stability and are scalable from 96 well plate format to 1536 plate well format. So they are perfectly well developed for screening and characterization applications, as well as lead optimization and head to lead sort of development work. Now moving to the working cell banks, these are high density vials and are manufactured to defined specifications, and we can develop them as Thorne-use format, where you take the vial, thaw it, and plate it directly in the assay plate and run your assay from there. And the applications for the working cell bank, you can use them for preclinical as well as for late phase applications, from screening, characterization, NAP testing to GMP lot release. So pretty versatile assay format we provide with our working cell banks, And we have over 20 years of experience cell banking. These cell banks can provide you a really potent tool to take your testing all the way from your screening step to your characterization and then finally to your late phase development. Now our flagship portfolio is our qualified bioassay. Qualified bioassays are highly reproducible assays and it contains all the reagents as a part of the kit. Our qualified bioassays are qualified with approved drugs based on ICH guidelines. In this case, we have qualified our assays, bioassays with Ozempic, which is a semaglutide, and also with Tirzepatide to show an ICH-based qualification that the assays have high accuracy, precision, linearity, and other attributes that you will need in ICH-based qualified assay. And I'm going to show you a case study for assay qualification in the later part of the presentation. Application-wise, the bioassays are perfect fit for late phase development, for GMP lot release, for NAB testing, and you can also use them in the initial phase for proof of concept work because they are provided as a optimized kit. Everything included in that kit that you can run assays and provide proof of concept data for your therapeutic. Moving forward, I just wanted to show you a bird's eye view of our portfolio for glucose metabolism. These include some of the receptors I already talked about, which are GLP-1, GIP, and glucagon. But in addition, we have developed assays for insulin, IGF-1R, GLP-2R, and amylins. So together, the entire portfolio enables our clients and developers to evaluate the glucose metabolism pathway in multiple different ways. And some of them I already talked about, including the 3 receptors, where we have qualified assays for multiple drugs in the pipeline. And again, the MOA-wise, we have assays that are based on cyclic AMP accumulation, beta-arrestin recruitment, receptor internalization, and calcium mobilization. And for other assays, we have the SHIP recruitment assays. And again, the assay formats available to you are in stable cell lines, working cell banks, and Thawing-use qualified bioassays. Now, for many years now, we are actively transferring our assays to global CRO, CDMO sites, as well as client sites for GMP lot release testing. And the assays I just talked about for GLP-1, GIP, and glucagon, we have already successfully transferred these assays to CROs and CDMO sites in multiple geographies. We have a really good system that supports assay transfers from our site to the client site or a CRO CDMO site for lot release testing for the therapeutic candidates for developers. So reach out to us to talk to us about our method transfer and assay transfer services from DiscoverX to your GMP, CRO, and CDMO sites. And we support global programs. Now let me spend some time on a case study for Tirzepatide bioassay where we show an easy assay implementation in lot release testing. And the case study is based on our cyclic AMP Hunter GLP-1 bioassay. Now to begin with, all our bioassay development work is driven by industry guidelines. We follow industry guidelines and requirements that are published by the regulatory agencies for cell-based assays to be appropriate to be implemented in GMP lot release testing. The key attribute for the assay is the assay should be reflective of the mechanism of action of the drug molecule. Secondly, the assay should be specific and stability indicating. Thirdly, the assay should show a relative potency range across at least 50 to 150%. So we accommodate this range in our assays to show the robustness of the assay across this wide range. Now we have used our assays and performed repeatability analysis, robustness testing, precision and accuracy to establish the assays are fit for purpose for GMP lot release. We have shown our assays are reproducible We have tested them as intra-lot, that is wild-to-wild testing, inter-lot, where we are testing multiple lots together, as well as looked at analyst-to-analyst variability. So collecting these kind of data sets enabled us to establish that our assays have the right setup to be implemented consistently in high stringent program like GMP lot release testing. We also are very mindful of long-term support, where our dedicated, well-characterized cell banks will help the consistent production of our bioassays across the life cycle of your program. We have supply agreements and we provide risk mitigation strategies as well as traceability, and we are always audit ready. Moving forward, let me show you a quick study plan that we have when we establish a qualification exercise for our bioassay. In this case, I'm showing the data for the Tirzepatide bioassay with our cyclic AMP bioassay. Now the study design, we have evaluated plate uniformity for positional variability and edge effects. This is an important evaluation that we perform to ascertain if the developer can use the entire 96 well plate, or they are going to be using only 60 wells, the inside 60 wells, due to edge effects. This is an important estimation which is required to ascertain the plate map for testing. And we have evaluated 5 nominal concentrations over a range of 50 to 150%, namely 50%, 75%, 100%, 125% and 50% nominal concentrations, with more than n is equal to 4 for each nominal potency testing. Now we have performed repeatability assays where we have done 4 runs at 100% nominal concentration by single analyst. So this data enables us to estimate the repeatability of the assay. Next, we have looked at intermediate precision for the assay, where we have incorporated 2 analysts running the assay across multiple days and multiple lots. And we aim for 100% success rate, where no plates failed system and sample suitability criteria in this assay. On the right hand side, in the table that you see on the top table, you see the dilution scheme for the assay. It is an asymmetric dilution scheme in which we have shown that such dilution scheme will enable fitting more data points in the linear range of the curve to make sure that the assay passes the parallelism requirements. Here is the plate layout on the bottom part, and different nominal concentration ran across different days and across different analysts. Now here we show excellent inter-day assay reproducibility data with Tirzepatide. Here we are running 3 different assays across 3 different days across 2 different lots. You can see the signal to background from this assay is pretty consistent with percent RSD below 15%, and the potency or the EC50 of Tirzepatide across these assays is below 10%, which shows an excellent repeatability of the assay across multiple days. Moving forward, here is the relative potency data, where we are showing a representative relative potency data across 4 nominal concentration between analyst 1 and analyst 2. As you can see, we have 4 different panels here, where 100% nominal concentrations were run against 50%, 75%, 120% and 150%. And the recoveries for each of these nominal concentration ranges are noted within the box. And you can see they are pretty close to what is expected from the nominal potency ranges. We have analyzed the data across 2 analysts in the table in the next slide. As you can see, the percent RSDs across the range is very tight, almost within 10% at all nominal concentrations, and the average recoveries are very close to 100%. This gives us an excellent dilution linearity data across the entire range, and the accuracy, repeatability, intermediate precision and linearity of the assay are showing excellent numbers. With this kind of a data, we ensure that the assay, when implemented in GMP lot release testing, will not fail. Our developers can use this data set to understand how tight the data looks like when they are going to implement this assay in GMP potency testing. Now, before I end, I would like to show you an additional development work that we have been involved in, creating our inducible cyclic AMP100 GLP-1 cell line. Now, talking about inducible cell lines, it's an important characterization tool that most developers will need to show that their drug molecule potency is influenced by the number of receptors expressed on the cell surface, because it's a cyclic AMP response and number of receptors do influence the drug potency. This is one of the requirements that we have seen many of the developers are being asked by the regulatory agencies. So for that purpose, we have created an inducible cell line for cyclic AMP100 for GLP-1 receptor. Here is the initial data that we have collected from this inducible system. The first panel here shows the GLP-1 expression using qPCR. As you can see, it's an inducible system which is induced by doxycycline, and upon addition of doxycycline across various concentrations, the expression profile of GLP-1 receptor does respond. It goes up as we increase the concentration of doxycycline. Now, in this assay, we have tested two different molecules, Exendin-4 and Tirzepatide. With Exendin-4 and Tirzepatide both, you can see the potency of both molecules shifts based on the amount of doxycycline added to the assay. Increasing doxycycline shifts the curve towards the left hand side, making it more potent, which is expected in a cyclic AMP assay where receptor density is supposed to influence the potency of the molecule. There is some leaky expression of the GLP-1 receptor even at no addition of doxycycline, which is also expected, but the relative potencies are highly right shifted. On the left hand side, the bottom panel shows the comparison between the inducible GLP-1 receptor which is expressed from a CMV promoter versus the one that is expressed using the inducible system for GLP-1R. And at 1000 nanogram per ml of doxycycline, it shows a comparable data that shows that the expression levels of the GLP-1 receptor matches the amount of receptor expressed from a CMV promoter line, which is a good reference point for us to compare the data sets with other cell lines that we have. Now, here's the data with the EC50s for Exendin-4 and Tirzepatide. As you can see, there is a clear pattern of increasing potencies as a measure of increasing concentration of doxycycline. And also to note that there is a clear potency shift for Tirzepatide versus Exendin-4, which is also well published in the literature, that Tirzepatide is a weaker agonist than Exendin-4 because it is designed to tune down its potency to take care of the side effects. And I will encourage you to visit our website, and we have a very comprehensive web page on obesity therapeutic panels that we have, the assays we have, and we have plenty of different case studies with ICH based qualification data for Ozempic, for Tirzepatide, and talks about our capabilities. We have several scientific posters. We have shown data for bias signaling and evaluation, and now we also have data for retatrutide. Retatrutide is a triple agonist for GLP-1, GIP, and glucagon receptors. And of course, we provide a lot of references where our assays have been published in the literature. So do visit our webpage to find out more on our assay portfolio for obesity therapeutics. Thank you very much for your time.